Good afternoon, everyone, to honorable judges and my fellow participants. I'm Miftahul Jannah from Faculty of Veterinary Medicine, University of Brawijaya, with my teams and under the supervision of Dr. Siti. Today, I will be sharing my research, a conventional PCR strategy for repetitions of tuberculosis in animal, primer, and protein design targeting gyrB. This is my outlines today. Tuberculosis, an aerosol transmitted disease, kills more people than any other infectious disease. A total of 80.2 million people were reported as newly diagnosed with TB in 2023. This is caused by Mycobacterium tuberculosis complex group. TB burden remains a critical global challenge to both human and animal. As we know, the species including in this MTBC group are various. There are human-adapted like Mycobacterium tuberculosis or animal-adapted like Mycobacterium bovis, primary in cattle, can infect humans via raw milks. TB has high zoonosis potentials if we don't have any strategies to control this burden, especially in Indonesia, which is the second-ranked country with TB burden in the world. Moreover, TB in animal is underdiagnosed yet poses a zoonotic risk in Indonesia. This underreported case in animals showed that just West Java, East Java, and Bali reported prevalent tuberculosis. There are two reasons why TB has almost high incidence cases. The challenges include show diagnosis and rising drug resistance. Standard treatment for tuberculosis requires at least 6 months of isoniazid and rifampicin. Fluoroquinolones such as moxifloxacin and levofloxacin are used as alternative drugs for these cases by inhibiting the DNA gyrase enzyme of MTB, specifically in gyrB subunit. Unfortunately, resistance to fluoroquinolone is increasing, mainly due to mutation in the gyrB mutations at quinolone-resistant determining regions. Thus, fluoroquinolones are now among the drug groups with rising resistance rates in TB treatment. The gyrB genes encodes B-subunit of DNA gyrase is an ATP-dependent enzyme that introduces negative supercoils into the double-stranded DNA. This gene is highly conserved within the MTBC, making it an ideal target for specific primer design. Early, accurate, and diagnosis in animal is crucial. Like conventional methods are limited by low specificity or long processing time. For these problems, we can use a molecular approach to rapid detection for tuberculosis. Thus, molecular diagnosis like PCR are needed. Faster and more specific, sensitive. Before doing this PCR test, we have to design the primary specific targeting for gyrB at MTBC group. This is the objective as follows. And for the methods, we conducted a sequence retrieval for NCBI database. For nucleotides, and we primer design with Primer-BLAST, we have evaluating the primer using NetPrimer and checking the specificity or MTBC with NCBI BLAST, and we have the optimum primers. And for the proteins, we sequence of using FASTA, and we building with Swiss model via this template and visualization of the protein of gyrB gene. And this is the nucleotides and the template that are using in this method. And the result of the study, we have 10 primers pairs, each 20 base pair were successfully designed targeting the gyrB genes of MTBC. The GC content ranging from 50% until 60% and the melting temperature between 50 degrees until 62 degrees. And among them, the 4 primer pairs were selected as the optimal due to its balance about characteristic and amplified product size and the physicochemical properties. The thermodynamic analysis showed acceptable flows of harpin, self-dimer, and cross-dimer formation with minimal complementary indicating reduced risk of non-specific amplification. And this is the predicted secondary and anomalies structure. This is the graphic visualizations of the optimum primer. For the 3D protein modeling, we have the secondary structure revealed a composition dominated by α-helices and beta-strands, typical of the DNA gyrase subunit involved in supercoiling activity. Specifically, interspersed with loop regions and strands that contribute to the flexible regions of the protein. The presence of both major secondary elements have a stable globular conformations consistent with the functional characteristic of gyrB. Again, this is the hydrophobicity surface mapping. Blue areas represent hydrophilic regions, while brown areas indicate hydrophobic regions, and the white one is neutral. And the surface is predominantly hydrophilic, suggesting that gyrB interacts extensively with the aqueous cytoplasmic environment, with total hydrophobicity negative 254.6. However, distinct hydrophobic patches are also observed in specific surface pockets, which may serve as active sites or ligand binding regions. And this is the ionisability surface mapping. Red areas indicate acidic residues like aspartate and glutamates, and the blue areas indicate basic residues like arginine, lysine, and histidine. The distribution of charged residues suggests electrostatic surface properties that may contribute to DNA or ATP binding, typical of gyrB's enzymatic function. The pKa total is 1661.7, show that the protein has ionizable and functional active residues. And for the conclusion is we have the primer with optimum characteristic and the 3D protein modeling, and the limitation of this study was fully in silico, and the recommendation is validating the design gyrB primer took in vitro and in vivo PCR assay. And this is for acknowledgement, and we can reach out the reference through this QR codes. And thank you. Hopefully these research can make a significant contribution to TB control. The circuit with Miftahul, Jannah, and my team.